Happy sunny Sunday morning. I hope you are all having an amazing weekend. I certainly had a great day yesterday and got to spend a little bit of time skiing, and I'm certainly looking forward to a lot of the upcoming busyness. I wanted to kick things off today with something that is so very pertinent to what we talked about last week. Remember talking about bacteriophages, and in fact, in one of the sections, I got a couple of questions about just evolutionary strategies for persistence within the bacteriophage population. And it's super interesting to find this article very, very recently published in Applied and Environmental Microbiology and then reported here by the American Society for Microbiology. And this is about a finding within ancient fecal material that they have recognized that certain bacteriophages were carrying antibiotic resistance genes. This is far before, so mid-1300s, this is far before the use of antibiotics as a treatment strategy. And so what we're recognizing here is a cool example of coevolution of bacteriophages and the enteric bacteria that they use as their host cells. And so remember that antibiotics are found naturally in the environment, and so if a phage is transmitting those antibiotic resistance genes to its host, it's actually giving the host selective advantage in living in environments where there are competitive other competitors that might be affected by the antibiotics that would be secreted by its host. So this is a rad article, and I think all of you should definitely check it out. I have retweeted it on my Twitter feed. That being said, we have a lot of busyness to talk about this week, and I am hoping that this Punctibyte will cover the weeks of the 3rd through the 7th, as well as the week that I'm gone, the 10th through the 14th. So let's get busy here. First things first, I wanted to say thank you so much for all that you did. The TAs like super showed out on Friday. I think there were all but a few of you who were there to help me with that incredible hecticness. We were just like so on top of things. It was amazing to have, 4 of you in 5028 inoculating unknowns at the same time as like 5 of you or 6 of you were out helping me with skills tests, all at the same time as we were allowing students to make up lab. So it was just an incredible example of our organizational team skills, and we were on fire. I mean, literally on fire. So thank you again, and I owe you all dinner. That's going to be like the first thing on my list when I get back from nationals. But speaking of nationals, this is the week that I leave. So notice that on Thursday there are 2 things happening. Thursday night I will be leaving town. Also on Thursday, our midterm grades are due. So in a moment, I'm going to talk about that. Sydney and Jonathan, you're up to cover Friday Open Lab, which I think also means Dwayne, and it also means Amy potentially. I don't know how that works. So I think it would be really fantastic if we could have 4 of you helping out Mara. Mara is going to be the one who's going to be supervising that day in my place. So it would be lovely if we could show out again the way that we did last Friday to really help her out. So if we could get 4 of you as would be the typical load for that particular section, that would be so great. Now, again, I am gone the whole next week when we're going to be working our way through Lab 16 and 17. And on that next Friday that I'm gone, Joe will be there for that open lab. Jesse, Dalton, Callie are all listed for that lab. And I guess if Callie's going to be there, Joe may not need to be there. So we'll want to coordinate that and make sure all of that as well. One note on that. On Fridays, one of the things that is on my schedule that goes off during Open Lab is my alarm to take out the buckets that would have been for the prior lab. So I'm going to ask your help in making sure that those buckets get taken out and put into the cold room so that they don't overgrow over the weekend. So that's something that you can help me out with and would be so very appreciated. So a lot of things that are kind of behind the scenes that need to get taken care of for all of that. The other thing that's going to be happening while I'm gone is Lab 17, during which we have our unknowns. So I'm going to spend a little bit of time talking about the unknowns, how to get the clues done for your unknowns, and to make sure you're all organized and ready to go for that particular lab. Only Erin and Erin will go on and start making wine in Lab 18 during the week of the 10th through the 14th. Okay, so let's begin, and let's first start by talking about midterm grades. So for the midterm grades, I've done a couple of things here. I have an Excel file that shows what I would like to be included on the midterm grades. So at this point, we've had, by my calculation, what I would like on the midterm grade are through pre-labs 1 through 13. But remember that the pre-lab for lab 10 and 11 was one pre-lab. So by my calculation, that should mean that we have 30 points coming from pre-labs. We then have had the unknown press release, 5 points, dilutions, 10 points, pipette man tutorial at 5 points, and the gram stain report at 15 points. This should tally to give 65. Now I know that some of you have more points than that already. That is, maybe some of you have already done the first notebook trek or will have done by the time we hit next Thursday. But I don't want you to include that in because not every TA will have done that. So particularly for those of you who TA a medical microbiology section, this is particularly pertinent. Those of you who TA general microbiology, all you need to do is to get the grades up to date in WIO courses. That ends your job. Med Micro, you all have a little bit more of a job, and that is I want you to download the grades from your Med Micro shell, because then that way I can make sure that the grades that I send to Dr. Monty are all out of 65 points. So here's how you will go about doing that. I'm going to bring in a sample gradebook from one of the Med Micro sections without student names. And I just want to show you that over to the left there is a button that's got the wheel on it and kind of the gear, I guess. And on that, if you click on that, there is a download scores. It's the second one down. It says download scores.csv. You're going to click on that, and that will enable you to get a download of your scores. It will open up in Excel. You can then save that in Excel. You can save this file as a .xls, and then you can make it possible to add this up to the points that we need to have for the 6 out of 65. And that's the file that I would like for you to send to me. Please reply to all on this Punctibyte if you have any questions or concerns, or you're just overall concerned about point totals and whatnot. Okay, so that is our job as come next Thursday. A lot of things going on, as I mentioned. Let me know if I can help in any way as you're doing that. Okay, now I think we should go ahead and talk about Labs 16 or Labs 13 through 17 and the TA guidelines for all of those. So beginning with Lab 13, we've already talked a little bit about Lab 13. It is the fairly in-depth Fungus Among Us lecture, and Erin Enriquez came in to me the other day and said, yes, fungus lecture successfully delivered. So Erin obviously did a lot of studying and did an excellent job of delivering that lecture. So congratulations to him. The rest of you still have that one up and coming. So this is a pretty in-depth lecture, particularly if fungus doesn't happen to float your boat. Therefore, as I noted, many of you have already gotten Tara to cover your lecture, or you're planning to have me cover your lecture. That's fantastic. If you're planning to do it, please use these TA guidelines, which do talk a lot about the different fungal divisions and about some of their characteristics. So use your guidelines wisely, and also feel free to do, and actually you should, do a little bit of extra research to make that a really rad lecture. That procedure is not very procedure rich, which may leave you a little bit of time to do some skills test on that day, and that would be great. Again, remember the skills tests are due the lab after spring break. The reason they're due that day is because I want to make sure that everybody has all the way through the Friday prior to spring break to complete their skills assessments. That is the last week. The week before spring break is the last week to complete the skills assessments, meaning that if they do their skills on that week, they only have one attempt at each skill. This has been working out really well, so I think we're having a bit of a win-win situation with skills tests. If they don't do them until the week before, it's because they've put them off. There's been lots of possibilities for that. Now, the next confusing thing is that we go straight from lab 13 to lab 15. The reason for that is that lab 14 used to be the practical exam. We now have the skills assessments instead, so you can just mention to your students that that's sort of like lab 14 is what they're doing with their skills assessments. So again, they can do those up through the Friday before spring break. Lab 15 is really also highly immersive and very, very engaging. It's all about the biochemical tests and all about identifying different bacterial unknowns. However, one of the very first things that we have to do is to establish profiles on groups of bacteria so that we know when we get a positive identification for something, we understand what that something is. It's just like with profiling of any type, you need to understand the culprit in order to be able to figure out the symptoms that might pertain to that culprit. So this lecture is very in-depth, and you'll notice that the TA guidelines for this go into far more detail than does the lecture itself. Students will just be being exposed for the first time, most likely, to the family Enterobacteriaceae versus Pseudomonasaceae. They'll just be for the first time thinking about major differences between the staph and the streps, the micrococci and the enterococci. And so if we can do a good straightforward job of presenting some of that, it's wonderful. But I would like all of you to have a little bit more background information and to understand a little bit more of the nuances behind these groups. For example, the staphylococci, one of the major things that we do to identify them is the catalase test. They are all catalase positive, as are all of the micrococci, but of course the staphylococci are much more likely to cause disease. Part of that is due to their virulence factors, coagulase being one important virulence factor, and that's the way we identify Staph aureus. But also remember that the staphylococci have the ability to lyse red blood cells, and in these TA guidelines, for example, we give the specific hemolysins that can be split into different groups and given Greek letters based upon the kinds of toxins that they are. So we see that you will find that there's more background to what we tell them than just, is it lysing red blood cells or is it not lysing red blood cells? So while we don't present every nuance to them, there are certainly more nuances. Same is true with streptococci, and there's a little bit more information given here. Obviously, we identify them as being catalase negative, but then there are many other things that help us to look at the difference between streps and staphs. Another thing that you may want to take time to look at is that amongst the family Enterobacteriaceae, I know I was talking to my group of TAs that is also my Canvas training group about some of these other members, because my the virtual world called Second Life, my name is Serratia, and I was reminding them that in fact Serratia is one of the many genera within the family Enterobacteriaceae. So just getting to know those a little bit more may be helpful to you and give you a little bit more of a sexy mind and maybe ability to converse with your students. Okay, so for Lab 15, I will still be around for Lab 15. So if you would like me to deliver that lecture, just let me know. However, it is a fun one. Many people really like to dork out with it, and I encourage it if it is something that interests you. Lab 16 actually is a lot of review. So after Lab 15 being very, very immersive, Lab 16 has a lot of questions that review the content from the prior lab, and it's pretty fun to deliver, pretty straightforward. The one thing, though, that you need to remember to do during Lab 16, and especially because I will not be there on that day, is you need to designate a TA to literally camp out at the oxidase station. Students need to know how to do the oxidase test, because if they do it wrong for their unknown, it will literally send them down the complete wrong path. So have one of the two or one of the three TAs camp out, sit back by the oxidase station, and show them how to do both a positive and negative control, and tell each and every group that they will need to do a positive and negative control along with their unknown when they do get their unknown. So this is highlighted in your guidelines here, and certainly something that will be a good idea. The other thing is that they will be inoculating KIA tubes on this day. With KIA inoculation, they'll take a stab, they'll stab down to the bottom and then streak the surface. And the hard thing about KIA is that it tests so many things at once. So remember that in the KIA tube, glucose is in excess, or glucose is in limited supply, lactose is in excess. So what happens is the cells that can eat both will burn through the glucose first, leaving only lactose. So what that means is that if the cell can use lactose, it will all stay yellow. But if the organism is unable to use lactose, it will begin using proteins. That is an aerobic process, which turns the slant red. So there's a common confusion for TAs in this test, and that is that there is no difference in the location in the tube where lactose and glucose are found. But what's different is the concentration. And the reason the slant turns when they go to proteins is because the incorporation of amino acids into catabolism is primarily aerobic. And that's why the slant turns red and not the bud. Okay, so I hope that that helps to clarify that a little bit. If you have any questions on the KIA test, please let me know, because it is one that is very nuanced and difficult to explain. Okay, that being said, we'll be able to go on to lab 17 and talk just a little bit of 17. This is a big deal because this is where they get their unknowns. So while the lecture itself is not necessarily highly nuanced, we do see this being a day where you need to be organized ahead of time. So you need to have your clues from your TA jump drive, from your TA folder. You need to have gone to that folder and printed off your clues ahead of time so that you can then cut them out and put them out on the bench along with the unknown tubes. Right now the unknown tubes have been inoculated, and I will put them into incubate early this week. We will then put them into their appropriate lab sections, and we'll want to make sure that we have the numbers that correspond with the tubes. There will only be enough tubes for the number of students in your lab section. So that means that you might need to make sure that your number of clues matches the number of tubes right before you get started. Let me show you on your TA jump drive so that you can see where you're going to find this. So look under unknown keys, click on that folder, and look at the unknown list fall spring. When you open this up and you go to the very bottom, you're going to notice that there are tabs on the bottom. This is the list of unknowns. You should print that. You only need your own section. But then you also will need to go to the next tab, which is the tab is labeled Spring Clues. So click on that tab. And then this will be what you need to print for your section. Now, remember, you don't need everybody's section, so just find your section and then print the numbers. So say that you guys are the TAs, say you're Wheezy, and you're in charge of this, and you're the TA for Section 12 General. You're just going to print just your numbers, starting with 241, and the clues for those numbers. Now, one thing I would suggest is just taking note of how these are formatted before you start printing. Make sure that the cells are large enough and that nothing has gone wrong with cutting off the clues. And then you can go ahead and print, and you'll actually be cutting these out. So you may want to make sure that there's enough room. Maybe you want to adjust these cells just a little bit so that there's enough room to cut them out and have those pre-cut clues for your students to pick up. Okay, so that covers a lot of the nuances for Lab 17. That's one of the big things to keep in mind and remember. The other thing that is going to be happening throughout all of this is that students are going to be looking at a lot of biochemical tests, and sometimes it's helpful for you to know and understand the kinds of issues that they might encounter. So first things first, on the hemolysis plate on blood agar, we will only, so for unknowns, we won't work with alpha hemolytic strains. We will work with some alpha hemolytic strains early on, and on their throat swabs, you will see alpha hemolytic strains. The difference there is hold it up to the light. If you can see light through it, that's beta. There are different degrees of beta. So the staph don't have as much light shining through as, say, strep pyogenes, the strep throat bacterium. But they're still beta if they're lighter than the instead of being darker, they're lighter than the rest of the blood. Darker would be alpha. And then if you just really can't make out a zone, that's when you start to say, okay, that's gamma. But it's important that you get well versed in that. Another thing that happens is with, say, the glucose broth tubes. Sometimes we'll see an orange glucose broth tubes. One thing to note is if there's a bubble in the Durham tube, that has to be positive for fermentation. So any bubble means positive. Now, unless somehow they've dropped the tube or they mixed it up and somehow there's a red tube that has a bubble in it because of that. So you just have to be careful that there may be some issues in reading some of those and educate yourself on what is going on there. The TA guidelines do help a little bit with that because they give some of the expected results. One note on the nitrate test. The nitrate test will be another one like the KIA that's hard to explain. Just make sure that you're careful to note that nitrate going to nitrite is detectable by the red color immediately. But nitrate going to nitrite and then to other nitrogenous compounds is when we see the colorless after zinc. So it's important to differentiate those two ways to be positive in your mind. My biggest suggestion is to please go to the virtual edge and watch my explanation of the nitrate test. It will help you to lecture on that particular test and to do a good job of explaining it. I'm going to end there. Even though Aaron and Aaron are going to go to Lab 18, I feel like we've already covered well more than enough, and I want to make sure that everybody does watch this. So Aaron and Aaron, if you would like to talk this coming week about Lab 18 in any way, shape, or form, because primarily it focuses on making alcohol, let me know. But otherwise, I think that's one that I know that you love to give and that isn't going to be terribly nuanced. I hope you all had a fantastic weekend. I can't wait until I'm able to have you over for dinner to thank you for everything that you've been doing, and particularly, like, you rocked my world seriously last Friday. It was like the most well-oiled machine ever. We are such an awesome team.